alumnos, a los 8 o 9 alumnos de nuevo ingreso a este programa de la maestría en Ciencias en Biotecnología Genómica. Al parecer no tenemos ingreso en el programa de doctorado en biotecnología. Si no me equivoco, doctora, no tenemos ingreso. No tenemos ingreso del programa de doctorado, pero sí tenemos el ingreso de los estudiantes del programa de la maestría en ciencias. Entonces a ellos les doy la más cordial bienvenida, siéntanse en su casa, sean bienvenidos y ojalá que los próximos 2 años sean de éxitos, de grandes logros en su vida profesional que hoy inician formalmente en este centro de investigaciones y de posgrado del Instituto Politécnico Nacional. Pues sin más preámbulo, quisiera, antes de que diéramos inicio formal, igualmente a este ciclo de seminarios departamentales que tiene instituido el Centro de Biotecnología Genómica desde hace ya un buen número de años, creo que casi desde el inicio del centro, hace 20 años, creo que hacíamos seminarios pequeños, recogidos, con poco personal, pocos alumnos, en una pequeña aula. Hemos ido creciendo a lo largo de los 20 años y bueno, esto ya se ha hecho una tradición en el centro. Han desaparecido algunos aspectos académicos, algunas reuniones, pero esto nunca desapareció, ha persistido a través de la historia del centro. Y ya es una tradición que tengamos nuestros seminarios departamentales cada semestre, donde cada uno de los laboratorios que conforman el Centro de Biotecnología Genómica presenta a través de sus estudiantes, de sus profesores, de sus invitados, profesores invitados, un tema que es propio del laboratorio, ¿sí? Dentro de la línea de investigación que desarrolla el laboratorio. Es la oportunidad para darse a conocer, es la oportunidad para que la comunidad entera observe los avances que ha hecho el laboratorio, los caminos, los desafíos, etc. En esta ocasión le toca el turno al Laboratorio de Biomedicina Molecular, del cual también soy el responsable, y lo va a presentar uno de los estudiantes que tiene el laboratorio. Es un estudiante de doctorado, es un estudiante internacional, viene de Nigeria y ya lleva con nosotros, en julio cumpliría su 3er año en el programa de doctorado. Acuérdense que el doctorado es de 4 años, ¿sí? Entonces él lleva ya un 70% de avance y va a presentar lo que lleva hasta el momento, le falta sin más preámbulo, pues le dejo la palabra al maestro Adebiyi para que nos presente su trabajo y después de ello pues tenemos una sesión de preguntas, comentarios y antes de retirarme solamente quiero hacer un anuncio. A las 3 de la tarde en la aula 2 tenemos la reunión con el sector de los estudiantes del CBG, ¿sí? Cada mes hacemos reuniones con cada uno de los sectores que conforman esta comunidad. Entonces la reunión con los estudiantes la programamos para el día de hoy a las 3 de la tarde. Todos los estudiantes politécnicos deberían estar en esa reunión para escuchar de primera mano y obtener de ustedes información acerca de la administración en curso. Los estudiantes de primer ingreso también, por favor, vayan a esas reuniones, ¿sí? Todos los estudiantes deberían estar ahí. Ok, eso es el anuncio. Los espero ahí a las 3. Los que no puedan por alguna razón, no pasa nada. Ahí nos vemos los que puedan. Ok, gracias.
Hello. Good morning. Good afternoon. My name is Adebiyi. I'll be giving a presentation on vector DNA barcoding in Mexico. What are the prospects and the challenges? To begin with, I will be showing us, this will be the overview of my talk. I'll be talking about vectors. First, to recap what we already know about vectors. The important one in Mexico, I'll try to briefly look at them and the species identification. To begin with, what are vectors? Vectors are organisms which transmit another pathogens to another. Vectors in themselves are not the cause of disease, but they are only like a carrier, and the pathogens need the vector to complete a certain aspect of their life cycle. If we all think of vector, there are three major ones that always comes to mind. You know, the mosquitoes, the ticks that we usually find on our pets, cats and dogs, and the sandflies. Vector-borne disease on itself are diseases that are transmitted by vector. They are caused as a result of being bitten by any of these vectors. But apart from these three common vectors, there are quite a number of them, and the WHO grouped them into 8 groups. And of all these groups, we have, of these 8, we have 7 of them here in Mexico. They are grouped into diseases based on their species and their life cycle and the disease that they transmit. Mosquito, we have here, that's the most common. We have the black fly that happens to not be no longer of public health importance because the disease that it transmits, onchocerciasis, has been eliminated due to the work of Dr. Mario. We have the ticks, which is more common because of our dogs and pets, and we have the triatomine bugs, that is the vector of Chagas disease. I quickly want to look at the epidemiology of some of these vectors, of the disease caused by this vector. So, and I'll be looking at mosquito now. Mosquito causes over 1 million deaths annually, of which 600 of the deaths are children less than 5 years old. In Mexico, here, the impact of this disease is not usually felt in the northern belt of the country, but if you move southward towards the tropical part of the country, like the Chiapas and other states downward, they're actually facing, malaria is actually of a public health significance. But dengue on the other part is endemic. Everywhere you are in Mexico, you are at risk of dengue, with about 2.5 billion people worldwide at risk. When you talk about epidemiology, look at the environment. Globalization and urbanization, climate change, temperature, everything has actually made the vector and the disease to spread to areas where they are hitherto not known to occur. So, controlling this disease, there are various measures to control disease, and part of them, which is very crucial, is vector control, of which there are major forms to control them. This is a chart to demonstrate to show us some of the world's deadliest animal. And the top 10 of deadliest animal in the world, of the top 10, 4 of them are actually vector. You have the mosquito as the leading, most dangerous animal in the world. You have the tsetse fly, you have the kissing bug, and you have the freshwater snail. Of course, the freshwater snail is not of public health significance in Mexico because here we don't have schistosomiasis. This is also a picture to show us the death, the spread of the death caused by vectors globally in a year. And in Mexico, we have between 20 to 50 deaths caused by vector. This chart is shown by, published by the Entomological Society of America last year. They're trying to show us that for vector control, there are three tripodial stands with which it is being looked at generally. They look at the pathogen, what are the cause of the disease, like the virus, the bacteria, or whatever. They look at the host, which is the animal that is being infected. And we always look at the environment. Where is the host found? Where is the pathogen found? What are the factors responsible for the pathogens? But most often in this discussion, the vector, who is the transmitter, is often usually excluded. And if we look at this set, at the center of pathogen transmission is like the tripodia, is the center is where the pathogen host and vector meet. Because if you look at the relationship between host and the vector, you have to know the host feeding to be able to know viral transmission, because obviously the vector feeding, because if the vector does not feed, obviously the vector is sometimes in epidemiology called an host, like the intermediate host. So because if the vector does not feed, maybe there will not be chances for transmission. And there is also the refractory individual environmental condition. What always brings the vectors and the pathogens together? How do they meet? Because obviously if the pathogen does not come in contact with the vector during feeding will not be able to transmit to the host. So at the center of all this, for you to be able to successfully control disease, you must be able to know what are the vectors. You must be able to identify what are the species of vector, where do they live. You must be able, if you identify them, it will enable you to study in detail the biologies of this vector. So you have to identify what are the methods that has been used to identify this vector. There are quite a number of them, of which I would like to group into 4 details. The morphological identification, which is the gold standard. It is what everybody knows. We have the crossbreeding, which usually deals with the idea of, okay, the species concept. Only 2 species we interbreed and produce viable organisms. If 2 organisms breed and they do not produce viable organisms, they are not of the same species. That is the basic biological concept. We have the enzyme electrophoresis and we have the molecular identification. But of all these, I'll be looking at the morphological identification, which is the gold standard. It is the gold standard, but it has its limitations, which are some of these. There is no doubt of it. There is skilled distribution of specialization of taxonomists. There is shortage of taxonomists. Taxonomists are the people that identify these things. We don't have it. They are inadequate. And there is lack of what we call standardized database. If you go to a museum or somewhere where the collection and a taxonomic collection, how a taxonomy A manages collection of organisms of the same species is quite different from how taxonomy B manage its collection, which might like of the same species. 2 organisms might have synonyms of the same name. Taxonomy A might decide to use a particular name for the same species of organism. Taxonomy B might decide to use another name for the same species of organism, which if outsiders that are not very familiar with the 2 names of the organism comes to like brought something for identification, might get confused using resources from the 2 organisms to identify the same organism. So this always creates a confusion. So then there comes this idea that was batted by a Professor Paul Ebert from University of Guelph in Canada in 2003. Please play.
DNA is a molecule that encodes the genetic instructions of an organism, and it is unique to each species. DNA barcoding is a method that uses an organism's DNA to identify it as belonging to a certain species. In Canada, our scientists are at the leading edge of DNA barcoding. At Agriculture and Agri-Food Canada, we have been identifying thousands of good and bad organisms and registering their DNA barcodes to international databanks. The benefits of this work and DNA barcoding are huge for our agricultural crops, livestock, and trade.
I work in mycology, and there are many cases where the organism would look the same in culture, they would look the same under the microscope, and we do need DNA sequencing to figure out if they're different species or not. Most Canadians would not realize it, but identifying organism is part of daily life. Well, you go to the supermarket, you want to make sure there's no pathogens in there. We did not import the plant quarantine organism with the food we're eating. So it is part of the daily life, and we don't see it.
With the Canadian Food Inspection Agency, we explore state-of-the-art technologies such as next-generation sequencing and metagenomics for the detection of pathogens in cereals and other grains. Accurate and reliable identifications of these microorganisms is paramount. DNA barcode datasets support the information provided on phytosanitary certificates and statements of assurance for grain shipments. These certificates and statements secure free market access for Canadian grain exports worth billions of dollars every year.
The challenges that DNA barcoding can resolve are really numerous. For my lab and our group here, we're really focusing on quarantine species, invasive species. One of these organisms can really cause hundreds of millions of dollars in trade disruption. So we're really trying to prevent the introduction, and we're also trying to develop tools to make sure that what we export is clean.
A recent example that comes to mind where DNA barcoding played an important role was a shipment of pulses to South America. By linking newly acquired DNA baseline data with specimen and other historical information, we were able to quickly confirm the absence of the pathogen in Canada, assuring compliance of the multi-million-dollar cargo.
The future is very promising for this kind of technology. The technology now is such that we're making it more available and easier to use by a wider range of people.
Okay, thank you. Okay, in that video, the lead scientist there made emphasize on rapid and accurate identification of pathogen, and he tried to let us know the importance where you can use it to quickly identify organisms, like to detect the absence of pathogen, like was done. That is, and you might want to wonder, well, what is the difference of barcoding, DNA barcoding, from the normal molecular identification that we used to know, that we have been doing? We all know about use of DNA and sequencing and all stuff. Well, what really tried to make DNA barcoding to be quite a little bit of different is that unlike the other molecular approach, DNA barcoding aims to lay emphasis on standardization. Like, okay, for all the identification, we are all using, for all species, for all non-species, we are using a particular gene. The normal biological identification might, like, okay, for my own culture, for my own this, okay, I'm using this system, I'm using this part of the gene, I'm using this primer, I'm using this PCR template, or PCR thermal condition, PCR condition. No standardization. Everybody use different things and different sequences. But DNA barcoding is trying to lay emphasis on standardization from the primer set that you're going to use from the gene that you are going to use, from the PCR condition, from the PCR component, trying to standardize everything and trying to standardize the process where you will go from data collection to the point of identification, standardize everything so that we can have, okay, this is this, we go for this, this is what we have. And it's trying to establish a database, a new database based on this standardization that will be used for future identification. But in all of this, mostly DNA barcoding has mostly been used for conservation of biodiversity. Okay, there's an invasive species or there's an endangered species we need to preserve. The use of it in vector control has not been on the front. And with the start of DNA barcoding, Mexico also established the Mexican Barcode of Life Initiative in 2005 with 3 major codes, in UNAM, ECOSOC, and SIBNOR. All of these nodes try to focus on different aspects of the 4th mega diverse nation on earth, which is Mexico, because Mexico has abundance of biodiversity based on their history. So this no, these researchers in this institute try to focus on that based on their own. But no much attention has been paid on vector barcoding, as emphasized by this publication, in which after 10 years of the establishment of the Mexican Barcode of Life, a review was done, and in their finding, of what has been done so far, none of it made mention of vectors. So now, I would like to look at some of the important, the prospect that we have for vector barcoding in Mexico. Triatomine bug, you might call it assassin bug, kissing bug, it has quite a number of names. There are about 31 species of this vector in Mexico. And this vector is responsible for the transmission of Chagas disease, or what you call American trypanosomiasis, that is being transmitted by Trypanosoma cruzi. Quite not so something that you can easily, there is no handbook or guide for how, if you go out there now and you see a kissing bug, how do you identify it that is specific to Mexico? We don't have any literature to guide that. You can only combine. And according to this research, this researcher, they try to look at the ecological niche, map the ecological niche of this vector in Mexico. And as you can see from the various maps, this vector, different species of this vector, you can actually find them almost in everywhere in Mexico. They can actually survive in everywhere in Mexico. That means that once you have a vector, you might likely know that you only need an infected person for a potential disease outbreak in that region. Okay, I'll also talk about black fly. Black fly is the principal vector of onchocerciasis, which has been eliminated in Mexico in 2015. And also, for this, there are about 31, they're not sure, no certain, no confident, sure scientific literature that you can say this is actually the number of black fly species in Mexico. There's depth of information on the systematic of it. We only know few about some of them that are actually responsible for the transmission of this disease. What else is out there, we don't know. Of course, ticks. This picture might be familiar to many of us that have pets. You could have seen some ticks, we see them, but there are quite a number of them. We have these are the hard ticks and we have the soft ticks. And they are made up of 3 families in Mexico. They are transmitting various diseases from virus to bacteria, and apart from being vector transmitting disease, ticks themselves are ectoparasites because they feed on the pet and derive them of nutrient. So, and unlike the earlier 2 mentioned vectors, they are actually something that can assist in identification of ticks in Mexico. But unfortunately as well, it is only limited to 1 genus, the genus Axodis. There are quite other genus that we have for ticks of ticks in Mexico. And the author of this paper concluded, like, to date, research on Mexican Axod has largely been conducted outside Mexico, with the result that most literature records are based on specimens in foreign collections, such as the Rocky Mountain Laboratory and the École Nationale Vétérinaire, which are in France, meaning that actually the Mexican research community by themselves has not really done much on knowing the diversity of this vector in their community. And the vector, still on the paper, the author concluded additional systematic research on Mexican ticks is urgently needed, not only to catalog this group, but also to augment regional studies of ecology, blah, blah, blah. More taxonomic elucidation of Mexican tick fauna will substantively contribute to investigation of zoonotic infections such as Lyme disease. The author as well trying to encourage more research into ticks by Mexican researchers, and he laid emphasis on catalog. How do we, in this modern day, with all the species and the time taken to go use the morphological identification method to identify species, how do we rapidly catalog the species of vectors that we have in Mexico if we do not use a reliable, and doing that correctly as well, without using a reliable technology. So sandfly. Sandfly also is another vector that transmits leishmaniasis. We have cutaneous and visceral leishmaniasis. These are also endemic in the southern part of the country, Campeche, Quintana Roo, Chiapas, and Yucatan. There are about 454 cases last year recorded about 2 years ago. Specimen identification, of course, like I said, is important for prediction and vector control decision, and there is increase in the geographical range of cutaneous leishmaniasis in Mexico. Also, on this, there is little that has been done in regards of using DNA barcoding to know how many sandfly species we have in Mexico. Some reports say it is 44. Some say, I've read some that said it's 50. We actually don't know the actual number of sandfly species we have in Mexico. So, and onto the last one, which is Mexico, which is the important that I deliberately put it to the back. In the North American region, Canada has a little number of species. Fact, we know that. Guatemala has 139. USA is said to have 147. They are, okay, how many do we have in Mexico? Some say it is the last bibliography of Mexican species said there are 124 species. 124. I've read another one that they said that there are 200. Some say it is 245. We actually don't know the number of species. And there are actually new species of mosquito that are actually not in there. I put this picture in here because I encountered this mosquito while I was working in the lab. I've spent about a couple of hours trying to identify it because that's the first time I encountered it. And I put it on Facebook and put the name like this or another genus. Of course, I was using the key, a guide that is not specific to Mexico because there is none. I was actually using a book that was made for identification of mosquito in Texas and southwestern United States. So obviously I got the identification wrong, and it occurs to me like, okay, if after, which of course, and that is what every taxonomist always make the wrong identification. If after spending hour in the lab using under the microscope, and I still cannot get the identification of one single species of mosquito that I'm encountering for the first time wrong. So what is the fate of the vector team on the field that are in charge of vector control? How are they meant to make identification? If at the end I just take the specimen and just do the molecular and go to onto the established database that is reliable, I can quickly make the identification. So, and this picture that I have here actually shows the importance, that try to elucidate the importance of vector control. In 2017, when there was the outbreak of Zika throughout the world, it was like a global epidemic. By the end of the 34th epidemiological week, there actually been high cases in different parts of Mexico. But because there is a prior idea of where the likely presence of this vector were, it was actually easy to map out the risk area based on the presence and availability of the vectors. This is just a key to show. This is one available kit to identify vector mosquito species in Mexico, but it's actually for the Anopheles, just one genus. There are a lot of other mosquitoes in Mexico that are not Anopheles. Anopheles are only the ones that transmit mosquito. The Aedes that transmit dengue, chikungunya, Zika are not captured here. And unlike the other countries in the North American continent, the Guatemala, and there's the key to identify, and Canada and US, they all have guide with which you can identify their mosquito. And as well, they have guide, and they have also been employing the DNA barcoding approach to actually identify their species. With all this, like I said, with all this, that mosquito does not have means of identifying their mosquito. There continue to be new record of mosquito species that has not even been previously reported in Mexico, because this is a report of 2018, and that is recording reporting another species of Mexico entirely new for the first time in Mexico, which means that maybe there could still be a lot of other species of mosquitoes out there that we don't even know that are in the country. So basically, this is just the method. This is just a schematic standardization of the DNA barcoding process from the sample collection that tell you everything that you need to do up to the region, to the step that we generate the DNA barcode. There's the DNA barcode there. Okay, so we did this, collect some sample, different area, we do the morphology and the steps. These are the steps necessary for the morphological identification. Then you do the molecular processing as described, like it's standardized, like I said earlier. This is the PCR condition and the thermal condition. This is the database where everyone involved in DNA barcoding are trying to submit that. This is database that is being constructed, that is being established for the future identification of all organisms, all life, plants, fungi, all life form. This is the database. It's linked to NCBI. The difference between this database and NCBI is that there are a whole lot of other bioinformatic analysis that you can do on NCBI, but this is solely meant for identification, and you can only find a limited set of genes on this database, not any gene. Okay, so this database, before we try to look, okay, what is available on this database as related vectors in Mexico, and I started with looking at the mosquito. This is what is found on this database for mosquito species in Mexico. These are about maybe less than 20 species. Some of them are even not, like these are not even identified to species level, only to the general level. And of these, there is not even any record for Aedes albopictus prior to before we start work. So, I'll continue. Okay. This is a tabular form to show what is there based on what we know, and the percentages are actually low. So after some period of work, we've been able to increase. This is what we have now from our own work. We have about over 50 species of mosquitoes that have been established, that we now have DNA barcoding for, that has now been uploaded to the platform to increase the use of the. We are just developing this platform for future use, but the use of this platform is dependent on the ability to be able to establish as much as possible all species, having sequence for all species that is present in a particular place. And when it is compared, when the database is established, any other person that wants to use the database doesn't have any worry. You have 100% assurance that once you get there, the result that you have is what you are trying to identify. And all DNA barcoding work right now are just the process of establishing that database, and that is what our scientists all over the world are trying to do. And we've been able to contribute record for over 50 species of mosquitoes from Mexico onto that database. And this is just like a figure to show this is what we meant before we start. The number of species that was in this database, the number of sequence, and the number of unresolved species. And we've been able to raise the number of species from barely about 20 to close to over 100 species for mosquitoes. Then for the sequence record from this, we've been able to raise well over less than revolving around 1,000 sequence for vectors and unresolved species. And also, this is, before we start, this picture shows the state where we have collection record from Mexico, from mosquito, prior to its start. But right now, we've been able to have collection from different parts, increase the collection from different parts of Mexico. But of course, there is limitation to what can be done. There, as you can see, there are still, based on resources, time, and so many other factors that has to be considered. There are still many places here in Mexico that are still yet to be covered. In terms of publication, because of course every researcher want to, the aim of every research want to do research and be able to publish. Not much has also been published as regard to this vector. For mosquito, for instance, these are only publications that you will find that is talking about DNA barcoding of mosquito in Mexico, like what we have, and they are using these publications. These publications are not even looking at DNA barcoding in itself. They are just using the gene, that the COI and the DNA barcoding and the COI gene in this their research. Their objective is not even DNA barcoding. This, I can say, is the only research that try to use DNA barcoding as an objective of their studying. And they are even only looking at a particular species, only one species, meaning that of the over 250 species thereabout of mosquitoes that we have in Mexico, there is still a whole lot of opportunity of research that needs to be done for mosquito alone in Mexico. So the same story also abound for ticks. There are only about 58 public records on this on the database, and only 56 of these are identified to the order level, not even to the genus level, only to the order. And they are only from these states: Chihuahua, Quintana Roo, Yucatan, Campeche, and this is the only species. And only from one institute, means that there is still opportunity for research using DNA barcoding on ticks in Mexico. No publication. There's no publication. Same thing with sandfly, no publication. Only 7 available public record for sandfly, apart from what we are doing that is not yet made available. And from this 7 records, 4 are only from 2 states, 4 from Quintana Roo and 3 from Chiapas, 2 species only. There are about over roughly 50 species of sandfly in Mexico. And this here doesn't even include the known vector for leishmaniasis in Mexico. So the same thing for blackfly. Relatively, a lot more has been done as regard blackfly, thanks to maybe some collaboration. There are 2 publications as regard that. A new complex, a new species have been done. Maybe it will be not too far-fetched to try to relate that to the elimination of onchocerciasis in Mexico. Maybe by the time we have the amount of work that has been done as related to blackfly, maybe some of the other diseases being transmitted by other vectors will have been eliminated in Mexico. Okay, this is the publication of blackfly, and on Kissenberg as well. There are only 82 available record, 26 sequences from those states, and also no record. No publication, only 12 species. Meaning that there are still opportunities, there are still more opportunities of what should be done. However, if you are looking at what should be done, there are things that we need to consider. The work of DNA barcoding can essentially not be done alone. It is a collaborative research, because it is like an interdisciplinary research, which as biotechnologists, you can only do the molecular aspect of the work. There is a need to collaborate with those that are seasoned taxonomists. Of course, so there needs to establish a strong partnership, a strong mutual partnership with seasoned taxonomists to be able to achieve and be able to maximize the opportunities that are in this aspect, new line of research. And also, in doing this as well, because part of one of the essential things of DNA barcoding is the storage of voucher. There must be an established means by which voucher collected for this work. There must be an established voucher collected for this work must be stored, or if they are already established specimen samples that have been collected, perhaps in Natural History Museum or wherever collections, there should be an established collaboration, understanding of how these samples can be accessed. It is until those are the challenges, until you are able to look for sustainable means by which these challenges can be overcome, then we will not be able to reach the maximum benefits, the maximum impact that this new line of research, especially as related to vectors in Mexico, proposed. In conclusion, my conclusion with generally is that there's still huge information gap that we need research to fill as we relate to DNA barcoding of vector here in Mexico. And this method is free. It is rapidly evolving. It is getting more and more accepted in the field of research. It is being used differently for every different plants, microbiology, everybody, metabarcoding. It is actually evolving, but not much has been done in the area of vector control. And there is a need for stronger collaboration and dedicated effort for substantial progress to be made in this field. Those are my presentation. Thank you for your attention.
Muy bien. Por favor. ¿Preguntas? ¿Nadie? Ok, Adeniran.
It is necessary to have the species in the lab to get your DNA barcoding, or you can do it by some bioinformatic tools using the already reported sequences.
Well, I don't know if I get your question right. Is it possible to? Yeah,
because well, maybe I'm wrong, but what I saw is that in order to get the barcoding, you need to select your species, extract the DNA sequencing, and then you have all this protocol that it is your barcoding. So it is possible to get barcoding
using already established sequence.
Yeah.
Well, the essence of DNA barcoding is for us to create a reliable database which we can subsequently use for identification. And if you look at NCBI, okay, everybody is just submitting sequence from NCBI. Oh, I see this is E. coli. I just say this is E. coli, and I sequence this, and the sequence I get, I submit to NCBI. Okay, NCBI, this is E. coli. Nobody really make a dedicated attempt to actually, maybe before. The person is saying, okay, I collected NCBI and I sequenced NCBI. How do you know it is NCBI, E. coli, and submit it to NCBI? And sometimes you find a situation where you will say, okay, from maybe that person is in maybe somewhere in South Africa. You are here in Mexico and you collected, okay, this is E. coli. You sequence, and by the time, okay, you call it maybe you call it Staphylococcus or whatever, and by the time you are sequencing your own, it is matching with somebody that calls his own E. coli. So what DNA barcoding is trying to do is that, okay, everybody accepted that for him vertebrate now, for vector now, this gold standard for identification is morphological identification, is the taxonomy. That is actually in little debate. So now let's bring these two fields together, the systematics, which is the morphological identification, and the biotechnological, the biotechnology aspect. Okay, let the taxonomy, the systematics, the taxonomy, let them do their identification using what we call type specimen. Type specimen are specimen like when this name, this scientific name that are recognized by IUPAC, when these names are being, okay, we are calling this particular organism, this maybe let's say Aedes aegypti. So there are people that their own line of research is that. So when they have identified that, okay, this, according to this social taxonomies, is Aedes aegypti, then you take a tissue from that identified specimen and use it to generate sequence, that specimen that is used to generate that sequence will be kept. Then you put this sequence, there is a code that you name this sequence, that organism. Then with the sequence, you now put it to that database, so that upon subsequent identification, if you bring your own sample, and it matches with this specimen, you'll be able, okay, this specimen is the one identified by this taxonomist, not just anybody that you don't know who identified. You have confidence, like this particular taxonomy. This work that I am doing, I'm not the one that is identifying. There is the expert on mosquito taxonomies in Mexico that is doing the identification. My own aspect of the work is to do the molecular part. I can identify, but I'm not an expert. Nobody can judge if I say this is Aedes aegypti, you say, are you an expert? I'm not I can say it is Aedes aegypti, I might be correct, but no, I'm not like an expert, I'm not authority in that field. So we need authority, the known authority, the limited taxonomic expert that we have in that field to actually do the expert. Then after they have done the identification, then we now use that, like create a link between the specimen and the person that identify and the sequence. Okay, this person identified this specimen, and this is the sequence. So you can use this sequence now to identify your own data. So in that, we might not be able to use sequence that already in the database, because we don't know who identified them. So we don't know how much we can rely on that.
Okay, thank you.
Alonso.
Well, thank you for the presentation, first of all. Just a question. I noticed from the maps that you showed that there are some states in Mexico that you already collected from, or I guess got samples from. But is it planned to continue with the rest of the states, or is that going to be just those for your, I guess, your thesis? Yes.
Okay. Well, like I said, the Mexican Barcode of Life is an initiative that has been around since 2005. Okay, I'm only coming in to fill a gap to contribute to the data. Obviously, with the schedule of time and resources, it is a project that will last maybe 4 or 5 PhDs. So I cannot do everything. It's not possible. So I'm only do my part to increase the level of information that we have in that regard. I cannot do everything.
Well, I guess in that same idea, do you know of any other groups that are like in the same you're sort of the same work?
Well,
from different states, maybe?
Yes, like I said earlier, this is not a research that you can actually do on your own. You need people to go to the field to collect the sample. It's like an interdisciplinary research. It hardly can be done on your own. Yes, I know of two or three other persons that are doing it, but I wouldn't know if it is enough for me to call them another group, because we actually work together. So I wouldn't know if it is enough for me to call them other group, even though there are aspects of the other work that they are doing that I am not involved. But I know that there are also some aspects of the work that we are both doing together. That they might be doing something else that I am not involved. But I still know that, okay, they are still doing something that I am involved. So I might not say they are not other group, like I will still call ourselves one-on-one, two-on-one, one research group, but not entirely the same research group. Like we have, like in our lab, they are not based here in CBG. There's another school. I don't know if you understand.
Yeah. I appreciate your answer.
Okay, thank you.
¿Otra?
Okay, I have this question from where he stopped about bioinformatics. I was just thinking that what's the prospect of the barcoding, in the sense that if eventually we have this missing gap field, if I choose to do my own DNA, then most likely it might also come to a time that we do the same thing in NCBI, that I might just need to just blast and see who is aligned to which. So I'm just asking, what's the prospect of this barcoding? Do you think it comes to a time in the research field that most of these species would have been deposited, and what we now have is just another person seeing something else that someone else has deposited over time? What's the prospects? What do you think?
Okay, thank you. Well, what your question is actually some of the issues that were raised by some of the people that were not willing to accept DNA barcoding before. But I would like to point it out, like, as soon as you have your DNA and you submit it to the database, there's what is generated that is called the barcode identification index, barcode identification number, bin. And this bin is generated by an algorithm that matches your sequence to sequence that has been earlier submitted, maybe from somebody else. So if by virtue you do your own identification and maybe you call, what is it that you're working on? What's the name of the bacteria that you're working on? And you try to call it something else. Maybe you are calling Delovibru and you are working on Delovibru and you're trying to call it E. coli. Well, I'll make it to you like somebody already worked on Delovibru from another part, and the person already has that tag, like the bin. This is the bin. This is the binary identification number for Delovibru. So when you are now bringing in your own Delovibru and you are calling it E. coli, it will not match with that bin that is already established for Delovibru. That means you already know that once you are not having the same bin binary identification number with all other bin for that species on that database, you know that your species that you are bringing is not the earlier mentioned species. I don't know if you understand me, if that is your question.
Ok. ¿Una otra pregunta? ¿No? Bueno, entonces agradecemos al maestro Adebiyi su presentación. Gracias.
Okay, thank you. Thank you. Thank you. Okay, see?